Hello. Hi. We'll start officially in a couple minutes. We're just going to wait for people to stream in after the break. I'm Dr. Shreejoy Tripathy, and I'll be leading the next session on for the KCNI Summer School. This is Day 3, Session 2, Transcriptomics at the single cell and bulk level. Yeah, so I'll just wait a few moments. If there's any questions from the session before, led by Dr. Dan Felsky, you feel free to ask them, and I'd be happy to take a crack at answering them. Or if Dan, I believe Dan is making some tea, so but when his tea is made, he might come back and and answer them. I think while we're waiting for people to stream in, I'm kind of curious to know what's the most. Maybe I'll just ask a poll. No, I think I will ask sort of generally. What's can someone sort of post in the chat and say what's the most interesting thing you've learned so far in the KCNI Summer School? Maybe I should ask a different question, an easier question, which is, have you felt like you learned something in the past, let's say 2 and a bit days, other than Dan making tea? Well, yes, other than Dan making tea. I mean, like something scientific or something about psychiatry or about the nervous system. Do you feel like you've learned, and do you feel like you're getting something out of the summer school so far? Docker, cool. The clinical talk on the first day was very new. Great. Yeah. Yeah, there was a lot of really great reception about the clinical talks. And I think that was something new about this year that we were pretty excited to try. And I think, yeah, it was a good idea. Anything else that people have sort of struck people in the session so far? Cool. Yeah, Docker. Docker's pretty cool. Yeah. 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 Okay, so I guess I'll just get started. So I'm gonna, let me start sharing my slides. Okay, share this. Honestly, each part was great so far. Well, it's very nice. I'm going to, if I hit focus screen, no, I actually want half. No, but this is good. So I will change the screen pretty soon. Okay, this is good. And then let me find my slides. And then, okay, and so I think I need to focus my screen again. So actually, hold on. I'm sorry. I'm stuck trying to figure out which screen is which. So just bear with me for a moment. Okay, so it's going to be challenging for me to see questions throughout the presentation, just because of the way my screens are set up. So I have a few places in my presentation where I'm going to sort of stop sharing my screen, and I'm going to try to answer questions as best I can. So please post if you have questions. It's probably best for everyone if you post them in as official questions in in Crowdcast, and so then we can, when I answer them, then I can sort of tag the question and it'll be posted in the in the feed, and then feel free to use the chat and as we have in the past, and I think that will be nice. Okay, so I will get started then. So let me focus this screen. I am screen sharing. Okay, and then let me go back to here. And okay, great. So you should be able to see my screen, and I will get started. So yeah, thanks everyone for attending the KCNI Summer School. We're on Day 3, Part 2, Single Cell Gene Expression and Integration with Bulk. So we are here right now, 10:45 EST, transcriptomics at the single cell level. As Dan mentioned before, I'm Dr. Shreejoy Tripathy. I'm the head of the computational genomics team here at the KCNI, and yeah, I mean, it's really exciting to be part of this summer school and exciting to be part of a integrative center that that goes from genes to gene expression to cells to circuits to behaviors. And there's other aspects too that are part of the Krembil Center for Neuroinformatics that aren't even on the slide, like psychiatry, and you'll hear about those throughout the rest of the summer school. I want to quickly mention that we'll have 2 workshops in the afternoon Eastern time. So the first workshop will be a workshop led by a postdoc in my lab, Dr. Sunny Chen, and that will be about an introduction to transcriptomic data types with a focus on single-cell transcriptomics. And the second session will be led by a new PhD student in my lab, Kian Arbabi, and that's on using transcriptomics data to help understand the changes in the changes in the brain in psychiatric disorders. Kian's an interesting example because Kian was I think a very integral part of the summer school last year as an attendee when he was a master's student at McMaster University. And so sort of in between last year's summer school and now, I've accepted Kian into my lab. We're really excited to have him start. He's been in the lab only a few months, and he's been become a very critical member to our lab. And it's really exciting that he's sort of gone from student to teacher. Okay, so just to sort of set things up, we talk about, we talk a lot about a lot of things at the KCNI, but a major theme in the KCNI is this theme of bridging scales of investigation of neuroscience. So, you heard in the past couple days about psychiatry in the clinical perspective and ethics in AI. You heard this morning about genetics, and particularly genetics applied to human populations. And those are just individual pieces of the puzzle of mental health. And one of the beauties and difficulties of studying the brain and the nervous system psychiatry is that there's many different ways of studying it, from genes to cells to behaviors. And we're really trying to help the field, or help psychiatry understand the brain and nervous system at various scales of investigation. And so we're going to hear, that's going to be a big theme of my talk today, and a big theme of the talks throughout the rest of the summer school. And so probably one of the central themes of the talk today is that there are many types of cells in the brain. So on the left, we have an image of, these are drawings of individual neurons that were drawn by hand by one of the fathers of modern neuroscience, Santiago Ramón y Cajal, who developed beautiful microscope techniques to be able to label individual neurons and visualize them under the microscope. And then so he would spend his mornings, he would spend his days looking at these neurons under the microscope, and then he would go back home and draw them for freehand in his notebook. So it's absolutely incredible that he could do this. And on the right, we have a more complex, or even more modern picture of cell type diversity, where we have all different types of neurons, different types of cells, and they're all sort of intermixed together in the brain. And this diversity of cell types, it's really great, but it creates a challenge in that unlike other tissues that are more simple, or more where the cell type diversity is not as great, like the pancreas or liver, the brain has so many types of cells, and it's where the field is trying to develop methods to understand the different types of cells. As Dan mentioned, there's this concept of the central dogma of molecular biology. So DNA is transcribed to mRNA, and that's translated into proteins. This idea, it underpins a lot of let's say biology. And it it was, I forget when this was actually developed, but probably in the 60s sometime. But it's despite, let's say, at least 50 years have passed since this was first posited as a theory, and it explains a lot of biology. One thing that's really important that will be sort of a theme throughout probably the rest of the day is the importance of mRNA. I think mRNA is really cool. I think I mean, I think that all of this is really cool, but I think mRNA is especially important, and mRNA is especially important as a way of understanding cellular biology. It's just one example of why mRNA is so cool. I believe everyone is aware of, you know, these new mRNA vaccines that have been developed to, you know, as effective vaccines against the coronavirus, you know, pandemic. And I think it's just so cool that, like, you know, just sort of explain how the vaccines work in case you're not aware. Like, scientists, you know, we have the sequence for the mRNA vaccine. They're able to package it inside these lipid nanoparticles. So when you get the coronavirus vaccine, what you're getting is actually these particles are containing the mRNA. And then what happens is that the mRNA then enters your cells, is translated by the ribosomes in your cells into protein, and then these proteins are then recognized by your immune system, and then that's what confers the immunity against the coronavirus to your body. And what's really cool about this is that, you know, this mRNA, it's literally just a code. It's a code of 4 nucleotides. And through, like, you know, inserting some of this mRNA into your cells, you can teach your immune system how to gain immunity against against viruses that cause pandemics. So I think this technology is really great, and it's going to lead to so many advances in medicine. I just can't wait for it. And again, maybe it's a bit hard to understand, or I'm not illustrating it quite so well, but this is because mRNA is so versatile. And it's really easy to obtain the sequence of these mRNA, and then you know, through this sort of the magic of these vaccines, the magic of these lipid nanoparticles, the scientific field has developed these vaccines. And so what I'm going to talk about today is how we can use this magic of mRNA to study cells and the diversity of cell types. So a lot of this is coming about through the magic and beauty of single-cell RNA sequencing. And so this is a relatively new technique that has, you know, caught cellular biology by a storm. We're currently in, basically this is an entirely new technique that didn't really exist even 5, 10, it's certainly not 10 years ago, barely 5 years ago, but now it's become like a benchmark method or a bedrock method of cellular biology. The basic idea is that there's many variants of it, but the basic idea is you can take a tissue from an animal or, you know, a human sample, you dissociate the cells, and then through a series of complex molecular biology, you can obtain single-cell transcriptomes. So here I've denoted that as RNA-seq, and I'll explain what that is. But basically, a transcriptome is a measurement of the abundance of mRNA per each gene in the genome. So, and through a series of methodological improvements, we've been able to obtain these single-cell transcriptomes for millions of cells in the context of an experiment. And rather than just measuring the mRNA abundance of a single gene, we can do that for all the genes in the genome. So it's this incredibly high throughput, incredibly high dimensional type of data that's now become relatively easy and routine. We are able to collect this type of data routinely. And then we can infer and then after you collect this data, then you can infer what cell types were measured by using computational techniques. And so you'll learn about that in the tutorials this afternoon. So I thought I'd just sort of take a moment to explain RNA sequencing. So as Dan mentioned, he mentioned DNA sequencing earlier in his session. So RNA sequencing is very similar. It's a way of it's a series of techniques, but basically it's a way of sequencing the RNA that's present in a biological sample, and then it reveals the presence and quantity of RNA. And it's rather than DNA, which is fixed per cell, and for most cells it's not changing. RNA is continuously changing, and it's a measure of you can think of it as like a snapshot of a cell or a sample in biology, at a single instance of time. So just to explain an overview of RNA sequencing. So So typically, if you have two samples, like let's say you have in this example here, you have a tumor sample, and in this example you have a normal sample. The first thing we do is we isolate the mRNA we isolate the RNA per sample. And then in the early days of the pandemic, the kits for isolating RNA were very hard to come by. So this is a tweet from a researcher, Michelle Monnier, I believe that's how to pronounce her name. But basically, a bunch of researchers were donating their kits for extracting RNA to biological labs so that they could use them for COVID testing. And then after you isolate the RNA, then we convert that to cDNA. So basically, that's turning these single-stranded RNA into double-stranded DNA. And then there's some additional steps of biochemistry. And then you apply with the cDNA, you then sequence the cDNA using DNA sequencing. So I'll explain what that is. But with modern DNA sequencers, we're able to sequence hundreds of millions of reads and generate tens of billions of base pair of sequence. So I will explain that right now through this YouTube video. Okay, so give me a moment while I figure out how to do that. Okay, so we're good here. I'm just going to quickly check my screen, and okay, great. And then here we're at this video. So I believe, hopefully I'm still sharing the screen, and you hopefully be able to
sequence of bases in DNA, samples are inserted into a sequencing instrument, where high-frequency sound waves break the DNA into smaller pieces that are only about 600 bases long. Special tags are added to the ends of the fragmented DNA. These tagged strands of DNA can then attach to a glass slide. In a sequencer, each piece of DNA is copied hundreds of thousands of times, which creates clusters of identical DNA fragments. Next, the sequencer reads the DNA, one base at a time, using different colored tags for each DNA base. Special sensors within the machine detect the different colored tags. This sequence of colors reveals the DNA sequence of each fragment. Powerful computers piece together these individual DNA fragments and reveal the sequence of your DNA. Then a team of medical experts uses specialized software to analyze and compare the DNA sequences to identify the handful of variants that may be important for your medical care. Discuss the result.
Okay, so I will stop there, and I will go back to my slides. Okay, yeah, so that's just a short video illustrating DNA sequencing. I think I just want to, again, it's kind of hard to illustrate how powerful this method is, but it's become incredibly cheap to collect this kind of data, and a lot of it, and well, yeah, so it's the first draft of the human genome, I think, cost $3 billion to collect. So one human genome, to sequence the entire human genome, cost $3 billion around the year 2000. And 20 years later, the cost to recollect the same data would be less than $1,000. So we've gone from $1 billion to $1,000. That's like 6 orders of magnitude cheapening of collecting the same type of data. And because it's so easy to collect this kind of data, and we can use the same technology for sequencing DNA, RNA, other types of epigenetic information from cells, it's become this incredibly versatile way of studying cells, genetic regulation, all kinds of things. And it's really a lot of this field, or a lot of the advances in this field have come about through advances in technology. So getting back to this presentation, so what we do is we obtain a nucleotide. The output of the sequencing is nucleotide sequences that tell us the ATGC sequence of each bit of RNA that's sequenced. Then teams of bioinformaticians like myself and the people in my lab, we then align these sequences, referred to as reads, to genomic coordinates. So basically here, this is a sort of like a genome browser based view of a cell. So this is a chromosome. These thick segments here, these indicate the exons. These thin segments are the introns. And so in each one of these segments, each one of these blue segments here denotes a read of mRNA that was then, or mRNA/cDNA that was then aligned to a specific place in the genome. And so we use sophisticated software to perform this this step called alignment. And then basically at the end of this, we end up with we count up the number of reads that are aligned to each gene. So for example, we count up the number of blue reads that align to gene 1, the number of red reads that align to gene 2, and the number of blue reads here that align to gene 3. We count those all up, and then we end up with a measure of the abundance, the quantitative abundance of mRNA per each sample that was put in. So for example, here I've illustrated this first gene, SCN1A. The y-axis indicates the abundance of reads that were aligned to that gene. And so we do that for each mRNA sample. And so sorry, it's kind of complicated, but I mean, that is what it is. It's a very high-dimensional, high throughput type of data modality. And so getting back to single-cell RNA sequencing, we're able to apply that same RNA sequencing technique that I just explained, and we're able to apply that to the thousands to millions of cells that sort of are profiled in these experiments. What most people work with when they, if you're not a bioinformatician, staff scientist, or you're not in a bioinformatics lab, but you're someone who's interested in working with this data, what you like the data type that you work with is this type of data called a count matrix. And so here on the left, I'm illustrating a typical gene expression count matrix where on the rows we have individual genes, so gene 1, gene 2, gene 3, and on the columns we have the samples that were assayed through the RNA sequencing technique. So for a single cell RNA sequencing experiment, each sample would be a cell. It's like cell 1, cell 2, cell 3. And the entries in the matrix indicate the abundance of each gene that was detected in each cell. So this 3 number here indicates that there were 3 reads of gene 1 that were obtained in cell 1. Okay, so I think I just got a Slack message, so I'm just gonna wait a sec. And actually, I'm just gonna go to my screen just to make sure that everything's still going. P0, sorry. I think we're okay. Can you hear the video? Yes. Oh, okay, that was me. Okay. Are we still good? Okay, I am going to just check my Slack just to make sure that I didn't run out. I think we're okay. I think we're good. Okay, going back to my slides. Sorry about that. Sorry. Okay, I can't really tell if I'm still on track. So I guess like someone in my lab, if I get kicked off, you can just text me and then I will know that way. Okay, so getting back to my explanation here. Right, so this is a count matrix, and so we have cells by genes. One thing I want to point out is that when you have zeros in this matrix, that indicates that you had no detected expression of a gene in a cell. And so this indicates the gene expression data. There's also what's called a metadata matrix that indicates per sample or per cell the information about each cell. So for example, here this indicates this is like each row is a cell or each row is a sample, and then this is cell 1, cell 2, cell 3, and so forth. And then this is information or metadata about each thing that was assayed. So if this came from a human experiment, we denote which human subject it came from, like patient 1, patient 2, so on and so forth. Whether that patient was in a control group or a case group, like someone with Alzheimer's, and the type of cell that was assayed. Yeah, and we're going to work with both of these types of data in the tutorials this afternoon. Just to, again, quickly touch on the point of technology, like there's been a lot of advances in the technology that have enabled this technology. One of the major technologies is advances in microfluidics. And so you kind of, I really like to show this. So this is an image of a microfluidics device where basically we have a glorified pumper here that's pumping out oil microdroplets. And the goal is to sort of obtain one of these big circles and one of these tiny, tiny circles that contains a single cell into a single microdroplet. And then in that microdroplet, you do all of the molecular bio. Oh, Okay, I believe my PowerPoint just closed, so I guess it didn't like that. It didn't like my GIF, so just give me a moment while I try to get that back up. Okay, no worries. Don't save. And here we go. Okay, and we're just going to go back to the other slide. Okay, thanks, Michaela. Yeah, it's a nice GIF. Okay, so I just like to sort of end, or before sort of moving on to, talking about why this technology is so great, just a few rules of thumb here. Like, one thing to keep in mind is that these technologies, they're only sampling the transcriptome, so they're not really measuring the entire complement of mRNA that's expressed in each cell. Most modern experiments that use these technologies are really only sampling, let's say, 1 to 10% of the mRNA that are expressed in each cell, and so by virtue of that, you're only really detecting the most highly expressed genes per cell. We're going to talk a bit about single-nucleus RNA sequencing, and one thing that I just want to mention is that so if you think about like cell biology, like mRNA is transcribed in the nucleus. So the nucleus is an especially rich source of mRNA, but the kinds of mRNA that you see in the nucleus are not quite the same that exist in the entire cell. And this is especially important for large neurons, like cells that have really complicated, complicated cell shapes. And lastly, this is more sort of a point for the experimentalists in the audience. Like, just like with all techniques in biology, they're very sensitive to the type of data that sort of goes into them. So this is very much like a garbage in, garbage out type thing, where it's very easy to spend a lot of money to collect this kind of data. It's very expensive to collect this kind of data. But if the samples that go in are garbage, you're going to get garbage data out. You're not going to be able to understand it. So yeah, that's just a quick word of caution. So in my lab and in many other labs, we make use of publicly available data sets that describe cell type diversity and cell type diversity in the context of mRNA. We really like the data sets collected by the Allen Institute. You'll hear about the Allen Institute for Brain Science throughout many of the presentations in the summer school. And they have really good, effectively cellular atlases of cell type diversity, both for the mouse brain and the human brain. We'll use the human brain data sets in the tutorials today. And one of the major data sets we'll use is this really great atlas of human cell type diversity collected through single nucleus RNA sequencing. And this, if you want to read more about this data set, you can read about it in a recent publication a couple of years ago. I'm not going to go through a lot of the details here, but this is just to say that using data from the post-mortem, both the post-mortem and the post-mortem brain and biopsy samples from epilepsy subjects, they were able to dissociate single nuclei from these samples and then profile the mRNA expressed in single nuclei using sophisticated techniques. And then they were able to basically define the various cell types that are present in the human neocortex, and then the mRNA profiles that characterize each cell type. This is a website where you can play with and you can find these data sets yourself, celltypes.brainmap.org. And I believe you'll get an opportunity to play with this website in the first tutorial this afternoon. And another thing that you'll do is you'll generate what are called marker genes. So these are genes that are specifically expressed in a single type of cell. So here these are my postdoc in my lab, Sunny Chen, who's leading the first tutorial, has generated this plot where he's used a computational algorithm to define marker genes that are enriched in one type of cell and not the other types of cells in the cortex. And so the circles here indicate both both the proportion of cells of a given type that express the gene, and the colors indicate how highly that gene is expressed in that cell type. And so these marker genes, they are very useful, and we use them for a number of purposes. And so for the tutorial, so the scientific concepts, really we just want you to become familiar with these types of data and learn about the concepts of cell types and marker genes. and gain familiarity with some standard steps of working with single-cell transcriptomics data sets. So I'm going to take a break for questions. I'm just going to stop my presentation here, and I'm going to check Crowdcast. Okay, so are there questions? So we're doing good for questions. I'm just going to wait just a moment before going on with my presentation. Okay, so I don't see any questions, so I will keep going. Okay. So one way that we use these single-cell transcriptomics data sets in my lab and in other labs is we use them to help understand how the brain is changing or has changed in the context of neuropsychiatric disorders. So, for example, there are brain banks that have literally brains in freezers. Scientists can then, microdissect out a small piece of that brain, obtain tissue, and then they can put that tissue in a blender and then measure the abundance of different types of mRNA in that tissue. And then again, they can use RNA sequencing to perform this experiment, where they're able to obtain quantitative levels of gene expression of each gene in the genome for a number of these post-mortem samples. And so we can do these experiments on control samples. We can do these experiments on samples with a neuropsychiatric disease, and we can ask, how are gene expression profiles different in people who have a disease versus people who don't have the disease? I want to mention that these experiments are largely done in post-mortem samples, and that's because, for many reasons, it's difficult to ethically collect samples of the human brain from living people. But I'll talk about at the end of my talk. I'll sort of talk about one situation where we can obtain brain samples from living people. And so, in my lab, we're really interested in understanding cell type diversity and how cell types might be different in the context of different disorders. Here's just an image sort of outlining some of the major types of neurons in the neocortex. Here, these are two major types of excitatory cells called extratelencephalic and intratelencephalic. The details aren't really important, but whether you're an extratelencephalic neuron or intratelencephalic neuron, that's defined by where the cells send their axonal projections. There's also a number of inhibitory cell types in the brain. I just want to highlight the cell type called SST cells that we'll talk about in the session and the tutorial as well. And so one thing that's really nice about this technology is it's become relatively straightforward to collect bulk tissue transcriptomes from hundreds of samples from these brain banks. But one challenge is that if we're trying to understand what's happening in two conditions, like condition A, it could be a normal condition, or a control condition, and condition B could be brains with people with major depressive disorder, or Alzheimer's disease, that if we see a gene sort of changed in condition B versus condition A, we're kind of left wondering, like, well, what might cause that change? So maybe that change could just be due to broad regulation, so that if you look at every cell in the brains of people with condition B, maybe you see that this gene X, is just downregulated. Or you could see that the change might be due to a specific just loss of gene X in a specific cell type, like these red triangles. Or you could imagine that it's due to a change in cellular proportions with no regulation. So you might just see that, well, if there's fewer of the red cells, these red triangles, well, that might actually result in why you see less expression of gene X in condition B versus condition A. And so in my lab and others, we've developed a number of techniques to help detect these differences in cell type changes in the context of different disorders using these bulk brain expression samples. So I'll sort of show you some an ongoing study in my lab and Dan's lab, led by Michaela, who is trying to understand cell type changes in Alzheimer's disease. And so, we know that there's broad, atrophy in the brains of people with Alzheimer's disease. So if you look at the brain here, you see that, clearly this brain with someone with Alzheimer's disease is different than this one. But we want to understand which cell types are changing most, and which cell types are changing most and which cell types are changing first. So we're using a technique that we call marker gene profiles, but basically it's one technique for what's called cell type deconvolution, and this is the bulk of one of the second tutorial that you'll do today. But basically the idea is that by using these single cell RNA sequencing experiments, we can define markers of specific cell types and then apply them to these bulk tissue expression data sets, and use that application to then infer how the proportions of different cell types have changed. So for example, here, these are cell type specific markers that mark dopamine neurons, and then applying them to bulk tissue expression from the substantia nigra from human control patients and patients who have Parkinson's disease. Then we can estimate we can estimate a relative proportion of each of this cell type, these dopaminergic cells in the substantia nigra, per patient, or so per sample. So that's what one of the dots here is. And then we can summarize the different groups. So we can summarize the control group and the group of people with Parkinson's disease, and then say that there's more or less of this cell type in patients with this disorder. And so Michaela has her project is trying to do this type of try to do this in the context of Alzheimer's disease. This is just one panel from her study where she's looking at a single cell type. These are layer 2-3 intratelencephalic cells. These are cells kind of up here that send their outputs to other parts of cortex and striatum. And each dot here is one patient sample from her study. And she has a number of data sets where she's looking at this, but by and large, she sees broad, down regulation or fewer of these types of cells in patients in the brains of patients with Alzheimer's disease. So these blue, on average, these blue circles are less than these red circles. And so we can do one thing that's really cool about transcriptomes or transcriptomics is that you don't really have to pick what gene or what type of cell you look at. So we can look at all of the types of cells in the neocortex, and we can ask which cell type is changing most and how are the changes. Yeah, we can basically compare. We can basically look across all the cell types and ask which is changing most. And so this is a figure showing that we see the strongest changes in this type of SST cells and this type of intratelencephalic cells in Alzheimer's disease relative to controls. So just again, to get back to this tutorial, you're going to learn how to use post-mortem gene expression data and infer cellular changes in the context of psychiatric conditions, and really the goal is to use these bulk brain gene expression data sets to say something about how the brain has changed and the pathology of brain changes in psychiatry. So just to sort of say a bit more about this method, so basically the way this method works is we're using cell type specific marker genes. I'm sorry for the sort of fuzzy quality on this image, but these are marker genes that mark a particular type of cell. Here, these are cell marker genes that mark the dopaminergic cell type in the substantia nigra. And in each column here is a different RNA sequencing sample. And so we're visualizing the expression of each gene in each sample. And we do that for a number of control samples, and a number of samples from people with Parkinson's, with a diagnosed Parkinson's disease. And what we do is so sorry for the complication, and this will be sort of described more closely in the tutorial, but we're basically taking these expression profiles, and then we're using the technique of principal component analysis to summarize the expression of multiple marker genes in each sample. And then we get a single number out, the first principal component that summarizes the expression of multiple marker genes. And so that principal component explains the dimension of maximal variation across the marker genes. So the assumption is that because these are cell type specific markers, the first principal component will summarize the relative abundance of the cell type in each sample. And then basically we turn that list of markers, let's say for here we have 10 markers for the cell type, we turn those 10 markers into a single number, which then we use as an estimate for the relative abundance of this cell type. And then we can compare that single number in across groups. So we can compare them in control groups and compare them in Parkinsonian groups. In patients who have diagnosed Parkinson's disease. And so again, this was a very quick overview of the method. You'll learn more about it in the second tutorial. So this is a good break for questions. So I will stop here and I will check my questions. So how do you differentiate if you are losing cells or if the genes are less expressed? Are they are they housekeeping genes? Yeah, so we do, Fernanda, this is a great question. So we have a number of methods for disambiguating that. So what we're doing in Michaela's paper or in her study is that, so the short answer is if you have just bulk expression profiles alone, it's difficult to disambiguate whether you have cells that are lost or you have broad downregulation of genes that are defining a type of cell. So what we're doing in the context of her paper is we're using an additional type of data where we can have a more direct measure of cell abundance. We're using single cell transcriptomes measured from people with Alzheimer's disease and control samples. And then we're also, so there isn't that much of that data, but using that data, we can directly just count the number of cells per type. And we're seeing very consistent, very consistent findings with that additional type of data versus these bulk tissue transcriptomes, where we're seeing in both experiments, we're seeing broad cell loss, or fewer of the cells that we're identifying, as opposed to broad downregulation of the genes that are defining each cell type. Yeah, but it's difficult to know, or actually it's difficult to say for sure. And really the best method would be to use histology and just count cells in tissue. So I think of these approaches as like, again, what's really nice about transcriptomics is you're measuring all the genes in the genome, and they're kind of like survey-based methods. And then once you can identify maybe the genes that are changing the most, then you would then study those genes, or so you would study those cell types more directly using dedicated methods like histology that require picking a marker or picking a cell type that you're going to study in the context of an experiment. So what did colors of circles? Yeah, so basically, yeah, Talisa, that's exactly right. So those are, so I'm going to say that I'm done answering this question. And yeah, so those plots, let me go back to that just to be clear. So this plot, this is what's called a group dot plot. And essentially the colors of the circles indicates the magnitude of the average level of expression. So when the circles, there's not, yeah, you're right, there's not a color code here, but the way this is coded is that red indicates higher expression levels and blue indicates lower expression levels. So the size of the circles indicates the proportion of cells of each type that express the gene, and the color indicates the average expression in that group. So high expression is indicated by large red circles, and low expression is indicated by small blue circles. Yeah. Okay, so I will get back. Are there more questions? Is the pace here pretty good? Or is it too slow or too fast? So there's no comment, so I will proceed at the current pace. Okay, so I will sort of just end, or sort of end, or go to the last part of this talk. So coming back to the slide that I initially started with, bridging brain scales. Another type of data that we use in my lab is data. So most of what I talked to you about is gene expression data, and gene expression data measured from the context of single cells and bulk expression samples, which sort of indicate which measure the mRNA from multiple cells in a sample. Another type of data we use is cellular physiology data. You'll hear about this type of data more in the next day in the context of the presentations by Dr. Itai Hai and Dr. John Griffiths. We use this type of data a lot in my lab as well, and I thought I'd talk about it. It won't be part of the tutorials for today, but I think it's important to talk about. So here's just an illustration of the collection of this type of data. This is often collected in thin brain slices. So basically, this method requires having living neurons, so they need to be obtained from relatively fresh samples. Like here, this is a schematic shown of a brain slice that's taken from a mouse brain or a rodent brain. And so by very carefully navigating a glass electrode to individual cells in these tissue slices, scientists can gain electrical access to the neuron, and then they can inject current into the neuron and then measure the cell's membrane voltage as a function of time. One of these lines here is an action potential, and we can measure all sorts of characteristics from that action potential, like its width, its height, and these electrical characteristics are another way of defining the phenotype of each neuron. My lab is very involved in trying to popularize or trying to disseminate a new technology called Patch-seq that combines this more sort of older method of characterizing neurons by their electrical properties, by their electrophysiology, and their morphology. The morphology is collected by injecting a dye into the neurons during these electrophysiology experiments, and then afterwards visualizing the dye using a microscope. And so the Patch-seq method combines these two more classical methods with single-cell transcriptomics, where at the end of the electrophysiology experiment, the scientist can obtain the nucleus and then put the nucleus into a molecular biology tube, and then send that tube off for what's now become standard single-cell RNA sequencing. And so from the same method, you could obtain the electrophysiology, the morphology, and the transcriptome of a cell. And it's really, this technique is very difficult, but it's very experimentally difficult, but it's really rich in that it's a really great way of connecting these different ways of describing and measuring a neuron. And it's really cool in that you can connect the transcriptome to these other more classical measures like electrophysiology and morphology. And so in a project in my lab led by a PhD student, Anikriti, where she's interested in looking at how patterns of gene expression give rise to the different electrical properties of the different cells in the brain, and really trying to connect the RNA to the electrophysiology features of each type of neuron. So I just wanted to end with a really cool, new type of experiment that we're pursuing in collaboration with Dr. Tawfiq Valiante and others. Dr. Valiante is a neurosurgeon in Toronto and at UHN and the Krembil Research Institute. And because he's a neurosurgeon, or sort of as part of his neurosurgery practice, he's an epilepsy neurosurgeon, and in a subset of his neurosurgeries, in order to treat the patient's epilepsy, he's able to obtain biopsies from the human brain. And so these biopsies contain living human neurons that can then be studied, like that would otherwise be medical waste, but then they can be studied using standard cellular neuroscience methods. And so a really talented research scientist in his lab, Homera Moradi, she characterizes the cells in the human brain using electrophysiology and morphology methods. So here's just a visualization of one of those morphologies, and by injecting current into these cells, she can then measure the electrical properties from these same cells. And so our collaboration is one of a few collaborations that's starting to collect this type of data, but it's really early days for these types of experiments, and we're just trying to the first question is just to understand, well, how are human neurons different from one another? How are they different in different people? Like might these neurons be different in the context of aging? Might they be different in the context of different genetics, like what Dr. Felsky talked to you about earlier today? We're really just starting to pursue these basic questions. It's really exciting. One thing you'll hear about tomorrow is using the data collected from these human neurosurgical experiments to build template circuits of the human brain in health and disease. And so this is an effort that's really led by Dr. Itai Hai, who's working with the data collected in Dr. Valiante's lab to use this data to constrain human basically brain computational simulations of the human brain that's constrained by this data. And here's just a one image that sort of comes from these types of experiments where Homera and I we basically just this just illustrates some of the diversity of the types of cells in the human brain. One thing that we sort of noticed when we were analyzing the data is that there's so much more diversity of cells in the human brain relative to what our expectations in the mouse brain. And so these are diversity among the different shapes of neurons, and these are diversity in the electrical properties as well. And so sort of bringing it all together, and what you'll hear about for the rest of in the context of the rest of the summer school, is how we can begin to connect these different types of data that you've heard about. So the clinical populations you've heard about the past couple of days, the different types of genomic sampling in terms of DNA, like what Dr. Felsky talked about this morning, RNA, like what I just told you about, and how we can begin to use that to understand how brain cells are changed, how different brain cells are changed, or how proportions of different brain cells are changed, and then use that information in the context of computational simulations and personalized brain models, which you'll hear about tomorrow from Dr. Itai Hai. And so this is very much like we don't have all the answers here, but this is something that we're all kind of collaboratively working together on at the Krembil Center for Neuroinformatics. So I just want to end by acknowledging my funding sources. We've been really lucky, or all of us have been very lucky to be funded to do this really exciting work. And acknowledge my lab. So we had this is from a lab social that we had a couple weeks ago. And that was the first time I saw many of my lab members in probably over a year. So it was really great. If you're interested to hear more about or see more about specific parts of our research, we have all our publications listed on our website, triplab.org. I didn't talk about a number of studies we're doing that basically looks at genetics and tries to relate genetics to some population features. Like, we don't have a study on almond milk and genetics, but we do have a study on how cannabis use interacts with genetics to increase or decrease the risk of psychiatric disorders, which is it's different from almond milk versus the genetic basis of almond milk preferences, but it's a study in kind of that vein. So you can find that at our lab website. Okay, I will end there and look for questions. So I believe we're doing quite well on time. So it's 11:37, but anyway, we'll look at the questions now. Topic is new to me. How does so let me start answering this question. So how does electrophysiology data look like? Does it change over time? Is it a single measurement per neuron? What are the challenges when integrating them with gene expression data? Oh man, these are great questions. I'm gonna see if Anikriti is around, and maybe she can answer this. So Anikriti, I'm gonna invite you on the stage. How do I do that? Give me a moment while I try to do that. Oh, okay, cool. Invite on screen. Okay, so I'm gonna invite Anikriti on screen, and then we'll give her a moment or two to join, and then if she doesn't join, I'll answer the question, and if she does join, we'll allow her to answer. Oh, and let me see. I'm gonna stop screen sharing. So close video, unfocus screen. So now you can see. This is good. Okay, so I don't see Anikriti here, so I will attempt to answer the question. So yeah, so electrophysiology data, does it change over time? Yes, it changes over time. And that's one thing that's kind of complex or complicated about any type of physiology data, is that it's dynamic. And that will be a theme of the talks, I think, in Dr. Itai's talk and in Dr. John Griffith's and Dr. Andrea Diaconescu talks, is like, physiology data is very dynamic. It's not static. Whereas if you think about gene expression data, or DNA data, it's static. It's measured at a single point in time. And so it's challenging to try to reconcile those two things. In practice, what we do is we collect features, or we featurize the electrophysiology data. So we try to define basic characteristics of the electrophysiology data, like the what is the cell's resting membrane potential, what is the membrane time constant, what is the input resistance, what is the spike width, so on and so forth. And then we basically obtain, just like we can obtain a count matrix for our gene expression data. Let me go back to that here. Just like we can obtain a count matrix for our gene expression data, we can obtain a feature matrix that describes the physiology data. So we have, rather than a gene expression count matrix, we have a electrophysiology feature matrix, but it's organized very similarly, where each column is a cell, and each row, rather than being expression of a gene, it's a quantitative feature. A quantitative feature that's measured in a consistent way across the different cells. And then at that point, it's just a matter of correlating the two or relating the two. It's probably not the most, that's one way of doing the analysis. And it's not necessarily the best way. And you're absolutely right that physiology is dynamic over time, and it's a measurement of living cells, whereas gene expression requires destroying the cell for the most part to measure that data. So there probably are other ways of doing the analysis, but yeah, what I described is typically what's done. That's a good question, Samar. Yeah, so Reza asks the question in the chat. I will attempt to read it out here. So sometimes a phenotype just give me a moment while I try to parse the meaning of this question. Right. Okay, so that's a really good question. So let me see if I can kind of summarize the question. So as I mentioned, when we measure mRNA, we're measuring a snapshot in time. We're measuring the mRNA from a snapshot in time. Whereas if you think about the cellular processes that are giving rise to cellular electrophysiology, well, when we're measuring physiology, we're measuring the output of proteins. We're measuring the contribution of a number of proteins that are largely living on the cell membrane called ion channels. And so those are proteins, and proteins have a longer half-life. I mean, again, to sort of use a term from physics, like, well, again, like when you're measuring mRNA, that's not the same thing as measuring protein. And so if you're measuring electrophysiology, you measure that, and that's arising from proteins and more indirectly from mRNA, then when you're trying to relate quantitative mRNA to protein expression or to electrophysiology, because of the disconnect between mRNA and protein, you might miss some associations there, and that's absolutely the case. So, but on the other hand, by the same magic of mRNA, we're able to measure we're able to measure the quantitative expression of each transcript in the transcriptome. So that's valuable as well. So I think probably, maybe I would sort of treat it as like a glass half full situation where, yes, we're, when you're measuring mRNA, there might be, there's turnover, or there's a very short lifespan, but then physiology is more long-term. But on the other hand, you're able to measure so many more different types of mRNA from a single cell. So yeah, so there's pros and cons to measuring mRNA. But again, by the magic of DNA sequencing and RNA sequencing, it's just much easier to measure many types of mRNA than it is to measure many types of protein from a single cell. So that's why I really like mRNA-based methods. But there are plenty of electrophysiologists who only study proteins. Oh, Anikriti just showed up. Hey, how are you doing?
Hey, good. Sorry, I was having some technical glitches on my end. Yeah. Should I still answer the question, or was it answered?
I think it was answered. Do you have more to say?
Oh, then I didn't catch it.
So Anikriti is a PhD student in my lab who is leading the project to integrate single-cell transcriptomics and single-cell electrophysiology.
Well, if anyone has questions, they can catch me up on GatherTown later.
Okay. Thanks.
Yeah. I'll drop out.
Okay. Are there more questions? There's a fair amount of time left. I could keep talking, I could say more about various studies we're doing. I think I'd prefer to answer questions or maybe end the session a bit early. But yeah, whatever the team, whatever the folks like. I have maybe a question for you guys. Let me do a poll. Yeah, I just created a poll. I'm generally interested in this question, like, are people interested in analyzing single-cell RNA sequencing data? Dan also asked if there are any questions from the first session that didn't get answered. Dan's available to answer them, and this is a good opportunity to do that. Dan, I can invite you on the screen, and we can do that. People who still have questions, feel free to post them here in the ask a question part, and then if there are questions, I'll invite Dan up to answer them. Twelve people are interested in analyzing single-cell RNA sequencing data, and no one is not interested, and no one said, huh. So that's cool. I think that's great. Again, like my background is in, I didn't really explain this. My background is in cellular electrophysiology. And I became very interested in transcriptomics when it became clear to me that I could sort of foresee the current revolution of single cell transcriptomics, like probably almost 10 years ago. Maybe, yeah, I could see it coming, and I was like, wow, this is going to be a transformative method to understand the physiology of cells. And so after that, I decided to do a postdoc in bioinformatics so I could learn how to analyze that data. And I'm very glad I did, because it's sort of enabled me, or it's enabled me to make use of all this data. It's really great. There's a question about Docker, and Sunny says you can use Docker, Binder, or Signet. We also have published Markdown documents for people who just want to follow along with coding. Yeah, we tried to make the tutorials very user-friendly, and you can interact with them at multiple levels, including just following along or running the code yourself using the Docker environment. Talisa asked a question, so I'm gonna invite Dan on screen so he can answer this question. Right, so Ali Reza, I'm gonna answer that question while I'm inviting Dan on screen. So Ali Reza asks, since the amount of transient mRNA is enriched in nuclei, is it better to use single-nucleus RNA-seq rather than single-cell RNA-seq? What other advantages each one has over the other? This is an excellent question. I feel like there's not really a good answer. So, and it's kind of there's pros and cons to each method. So on the one hand, single-cell RNA sequencing may or may not be advantageous because it contains mRNA transcripts from more of the entire cell, in particular mRNA transcripts from the cytosol, whereas single-nucleus RNA-seq just would have RNA from the nucleus. So that's one sort of pro against single-cell RNA sequencing versus single-nucleus RNA sequencing. But single-nucleus RNA sequencing has a number of advantages that are maybe has a number of advantages in that it's effectively impossible to dissociate single cells, intact cells, from frozen post-mortem tissue. But for whatever cellular biology reason, it's possible to do that dissociation of single nuclei from frozen tissue. So basically, single-cell RNA sequencing is effectively impossible for most human samples, except for a few rare cases where you can get fresh human brain tissue, like the epilepsy surgery experiments, like I just told you about. What other advantages each one has over the other? Yeah, so those are the two major advantages. Also, seeing one sort of, Anikriti, you're still on the screen. Are that intentional? Okay, I'm gonna kick you off. Okay, so what other advantages? And then one other advantage of single nucleus over single cell RNA sequencing is that the act of dissociating cells from tissue can induce some stress on them, and single nucleus RNA sequencing tends to be a bit more have less of that bias. So basically, there's no perfect method, and it's really dependent on the specific situation that each person might be in.
Yeah, it's harder. It's a lot harder to keep a cell intact than a nucleus.
Mm-hmm.
And if you think about tissue samples that you may have, frozen samples, for example, will not be amenable to single cell RNA-seq because you have leakage. You lose the cell membranes. So you can there's some advantages there. I mean, generally the idea is it's tough. There's so much that you miss. I mean, as Shreejoy kind of pointed out, there's so much that you miss, but also some of the complexity is hard to make sense of at the full cell level.
Yeah, I just want to mention that it's sort of, having been in this field for almost a decade now, it's wild how fast it's moved. And only a few years ago, single nucleus RNA sequencing wasn't really a thing, but now it's sort of become the industry standard, and just the technology is moving so rapidly. And so certainly it's possible in a couple of years or two that maybe single cell RNA sequencing will be more standard. So I don't know, we'll see. But right now, I think at least for working with archived frozen human samples, the default is single nucleus RNA sequencing. Dan, do you want to try to address some of the questions? When you start answering, maybe you read the question and I'll click start answering for you. Do you want to answer Talisa's question?
Sure. Yeah, great point. So Talisa's asking about the issue of multiple testing correction when calculating and testing polygenic scores using a clumping and thresholding method where you have to choose p-values fairly arbitrarily. This is actually something that I've thought quite a lot about. I've got a couple students who are working on this at the moment. And other papers who have compared iterations of clumping and thresholding have thought about this too. So papers have done really weird things like develop composite PRSs from a set of polygenic scores of the same summary stats generated at many, many p-value thresholds. So there was a paper, I forget what they called it. It wasn't meta PRS, it was like stacked PRS. I can find the paper for you. They basically just, they encountered the same issue that you're kind of thinking about, So if the fundamental problem is if you calculate, let's say 10 polygenic risk scores for major depression at different p-value thresholds, they're actually not always going to be very well correlated with each other. When you include very different SNPs, you can actually include different types of information based on where in the genome they are and how strong risk factors they are. So the optimal predictive p-value threshold for a given score might be different in different samples for different purposes. And as you expand on that, as you look at 100 p-value thresholds, 1,000, 10,000, the issue can become even harder because what's the solution, right? Do you test all of them, or do you only kind of pick one at random? Do you just take genome-wide significant variants? I mean, there are some GWAS with no genome-wide significant variants, and those are always changing as we update the summary stats. So you can calculate like hundreds or thousands of polygenic scores at different p-value thresholds, combine them in your analysis using something like a Lasso regression or some kind of dimensionality reduction technique like PCA. Basically try to capture as much of the variation along those, in all of those scores as possible, or generate some combination of them that creates one kind of predictor variable. And we find one of the papers that does that. The other one, which is the most common way, which is outlined in the paper that I mentioned before, is that you have to use the ideal way to do it objectively is to have a tuning sample. So for example, if you're interested in testing a polygenic score for some disorder in a new sample, and let's say your new sample is a big sample, let's say you've got 100,000 people, the thing to do is to separate out 20,000, 30,000, maybe not that many, 10, 20,000 people from your sample, do basically a cross-validation experiment. So it's all about, this is like a common theme in machine learning enterprises and model selection. It's a question of model selection. So if you have 10,000 polygenic scores to choose from, which one do you choose? Well, you have to have a second training sample, in essence, that you cross-validate and identify the optimal predicting score. And then that score is the score that you carry forward into your actual analysis on unseen data, on new kind of data sets. They refer to it as a tuning sample quite often, and clumping and thresholding is one that typically requires a tuning sample. Really hard to do if you don't have large numbers. So for example, if we're trying to evaluate the effect of, like, I mean, it's one of my students is doing, Irvin Teo, as well as a friend or another student, Amin Karagani, who's actually a TA. Yeah, if you've got like 2,000 people, we're looking at polygenic scores calculated from GWAS stats in the UK Biobank on like 500,000 people, and then calculating the scores in 2,000 people who have like post-mortem data. It's very difficult to do a model selection and like a model for the purpose of inference in that same set of samples. You just don't have the number of subjects. And of course, because polygenic risk scores aren't really that specific and aren't that like profoundly powerful, depending on the disease, you do kind of need numbers, yeah. But the tuning sample is usually, I don't know what the ideal kind of, sample size is for the tuning parameter, but you can take a look. So the one that I'll post the papers in the chat.
Okay, I'm gonna annotate done answering for this question. Yeah, Talisa, that's a great question. Dan, I think there's a couple more questions that have popped up.
Yeah, about including different ancestries in GWAS and how it can actually increase power. Can you elaborate, and is there a good method for doing this? Yeah, so that's a good question. I'm not an expert in multi-ancestral GWAS and the kind of guts of how they modeled it, but there's a paper that I will post after I post, just making a note here, after I post about the benchmarking studies for tuning samples in PRS. I don't actually think, so it was a paper where there's a new cohort for evaluating complex traits. It's a very large GWAS cohort of kind of mixed ethnicity samples. I forget the name of the consortium. I'll post it. But basically, the authors did a full GWAS and PRS analysis using each sample independently and then together in mega-analysis and also in a meta-analytical framework. And I don't know if they did anything beyond just kind of combining them or if they treated them in any separate way. So I can post you those papers. I mean, the intuitive explanation is that in the fine mapping, if you only have one ancestry, then you only really have one set of haplotype structures to rely on in your mapping. And that's important because you may have seen this in the locus zoom plot that I showed. You have a credible set. This is basically like using LD structure, you can come up with like a credible set of SNPs that might include the causal variant. And that calculation relies on estimates of LD. And if your reference data set is 1,000 genomes European, or it has to be matched to your GWAS sample. So if your GWAS sample is not mixed, then that LD structure, that credible set, won't actually include SNPs that might be causal. So it expands your credible set, and it also can do the opposite. So if you have, for example, two haplotype blocks, that one's present in one population, one's present in the other, and your test statistics are revealing linked SNPs, not causal SNPs, then actually in the cross-ancestral GWAS, your kind of credible set analysis will only show overlap, right? You'll only actually extract signal that is kind of confidently associated in both of those haplotype structures, and that boosts the signal. I'll point you to the paper that it does a better job of explaining it than I do. But needless to say, when explained correctly, it's quite compelling.
Dan, is the cohort you're thinking about the Million Veteran Project?
It's not the Million Veteran Project. No, it's another one. It's another one. It'll come to me.
Okay.
They combined it with the giant consortium data, and it's a consortium specifically kind of came together for cross-ancestral analysis, and it is it's really good.
I know sort of anecdotally, like we're experiencing this a fair amount, like in the work in my lab that uses GWAS summary statistics. Like our preference is to only use them on subset the sample to just European or Caucasian participants, white, anyway, whatever the PC term for white is these days, but it's becoming increasingly less. Like reviewers are less okay with that, or the public is less okay with that, and we want to make sure that the studies we're doing on human genetics are applicable not to only white people. And it's awkward as like not a white person to do a study that's only applicable to white people.
Right. Yeah, I think that the issue is that now, like, there are data sets that you can look at, like UK Biobank, and Talisa's pointing to this paper from Ben Neal's group looking at like multi-ancestry GWAS. They've made available really detailed, nice explanation of how they went about doing multi-ancestry GWAS in the as part of the Pan UK Biobank Consortium, which is kind of like version 3 of Ben Neal's GWAS analyses in UK Biobank. And it just shows that like it's all in UK Biobank subjects, and it's not huge compared to some of the sample sizes that large consortia that are disease focused can gather in terms of cases and controls. But there are tens of thousands of people in that UK Biobank sample with non-European, Caucasian, or British ancestry. So if you're doing an analysis in UK Biobank on genetics, there's no excuse. That's the thing, is you're doing it like an association analysis. You may be able to justify, depending on what your outcome is. You don't have enough power to look in tens of thousands or smaller if it's a specific diagnosis. But the data are there now, and increasingly so. The All of Us consortium is like UK Biobank for the United States. They're going to recruit 1 million people, and half of them are going to be non-European, non-white, essentially. So it's supposed to be more representative of the United States population. So at that point, like, if you can look, why didn't you? That's kind of the idea. It's a bit more complicated when thinking about designing an actual concise or like a compiled analysis where you incorporate them all together. Because as soon as you start dealing with multiple ancestries, so multiple homogeneous ancestries is one thing, and you need to, your imputation is affected. The way that you kind of handle that analysis is also affected. But with certain populations, you also have admixture, which is like a major thing in South American populations, Brazilian populations, Caribbean Hispanic populations, where you have the influences of multiple ancestries together. You can't use standard methods to work with that data.
Right. Dan, do you want to post a link to your Caribbean Hispanic paper?
Yeah, I mean, it's not a good example because it's not a GWAS, but yeah, it's a transcriptome analysis. I can actually post a paper that we just published, the first polygenic risk score for Alzheimer's disease in Caribbean Hispanics.
Yeah.
We just published that in Annals of Neurology, so I can post that too.
Yeah. I'm going to take a moment and answer this question that just came up in the chat from Ali Reza. So it's basically a question about biology wet lab methods for collecting single cell transcriptomes. And the gist of the question is it better to use commercially available methods like the increasingly popular 10x genomics method for collecting single cell genomics, transcriptomes, Patch-seq, whatever, or is it better to use more sort of like, let's say like DIY, do-it-yourself methods, like she mentioned, or this person mentioned split-seq, combinatorial barcoding to profile single cell transcriptomes. So I should just say I'm not a wet lab scientist. I don't work in the wet lab, but I work with many people who do. I think my answer for most people would be use the commercially available method. They tend to be a bit more expensive, but per sample they tend to be more expensive, but you're more likely to get usable data out than attempting a more do-it-yourself method like split-seq. But clearly, if your lab has a lot of molecular biology expertise and you have expertise using very new methods that might require a bit of optimization yourself in the lab, then by all means go for do-it-yourself methods like split-seq. Yeah. Dan, I think there was one more question from Samar about recommended pipeline for QC imputation.
Yeah, well, great point. Yeah, so the UK Biobank did their own imputation, and I think they did it using the Haplotype Reference Consortium. One of my students, someone can correct me if I'm wrong. It's not hard to find on the UK Biobank like data release. They've done an imputation. It's a little. So for virtually every consortium, except strangely for ADNI, if you work with Alzheimer's disease, it's a bit frustrating. They typically handle imputation on their end. If they're collecting genetic data, they'll have experts on multi-ethnic imputation, multi-ancestral imputation. Sometimes it's handled in a more sophisticated way than in others. Sometimes you just impute them separately, sometimes all together. Now I think the recommendation is, TopMed panels, the TopMed panel is a diverse panel. It's kind of including multiple ancestries in the same reference panel can also help imputation quality, oddly enough. So you've got one thing, Plink2 recommends that we use a relationship pruned sample ID list, which comes with UBIC, right? So basically, this comes down to like the idea that most people, when running these genetic analyses, will be stratifying the populations, as you should. We saw from my polygenic score analogy or kind of example that will skew your results in a big way. So what UK Biobank has done is they've generated this principal component analysis, this kind of ancestry plot, and separated out people who kind of map onto different ancestries as established by, I think, the 1,000 Genomes Project. Almost exactly like you saw me superimpose the Chicago sample, this Ross map sample, onto that projection of ancestry. So they've done that already and assigned each person in the UK bank to some ancestral group. So they've done the work for you in terms of looking at the raw data. You can, and they've also, so we're relationship pruned. They've also done an analysis of cryptic relatedness, of basically how distant or relative are people to each other. They can identify that because, again, at the population level, if you're looking across the UK, or at least many sites in the UK, you might just have like cousins, second, third cousins, somewhat distant relatives in the analysis, and that can bias things. So you may want to get rid of any people just at random, one person who's related to another person. They've also I believe they've done that. They also may have generated a kinship matrix, which is used in these like mixed models for GWAS and things. A matrix that basically shows how each person is related to each person, which is a huge amount of data. So this leads to kind of answering your question. Plink2 is pretty good with dealing with really large data sets. It's better than Plink 1.9 was, but it's still not as good as some software like Ben Neal's group uses HALE. They developed this program called HALE that uses specific kind of optimizations for matrix algebra to run like gigantic amounts of data, because it grows exponentially when you have more variants and subjects. Every time you add another subject, it just blows up. So HALE is what they used. That's what they used in the Pan UK Biobank Consortium. And it's also optimized for cloud-based work. They've made their kind of, I think a lot of their analyses were actually performed on the Google Cloud, like at the Broad kind of works with Google Cloud. They have their own server farms, but I think that they've optimized it for working on the Google Cloud, and a lot of the data is also like hosted on Amazon Web Service. Yeah, just a GCTA is like, very like the standard. These are all like the big tools. But I don't know about, for imputation. So if you're doing QC and imputation, I guess actually I didn't really answer that for imputation itself. I don't know how they would manage a full-blown imputation on such a large sample, other than just breaking it up into pieces and running it in parallel, or because they didn't use the TopMed panel, and I don't think they didn't submit UK Biobank, I think, to the TopMed imputation server, although they may have partnered with Michigan to do that. And there's no shortage of experts in imputation at the Broad. They probably just had their own in-house pipeline, but we'd have to read about how they did it. Because it's such a large data set, it's really not worth running imputation on your own, because the raw files are so large, and the reference data for TopMed is huge. It's why it's all hosted on the cloud. But yeah, I would use what comes with the data set in almost all cases. Sejal, who we heard from on Monday with reproducible computing has gone through this with a Canadian study, the Canadian longitudinal study of aging, only 30,000 people. And dealing with all of that is, oh, you tried that. What is it that you tried? The CLSA data, or you tried imputing your own data? The UK Biobank?
Probably.
Did not work, of course. Tried imputation. Yeah. Doing it on your own can be an enlightening experience. But that's best to leave it to the cores that have the pipeline set up, for sure. In smaller cases, do it yourself and do it on the TopMed imputation server. It works just like the original Michigan imputation server, Sanger imputation server, where they had HRC and 1,000 genomes. They've optimized this. They've done some optimization of the QC that's necessary to upload your files, because you have to do some quality control on the raw data before you upload them, and you have to put them in like a zipped VCF format. Just as long as that's done correctly, it's pretty okay. And what they give you in the end still requires some annotation, as Sejal knows, because they give it to you in chromosome and base pair position format rather than just putting in RS numbers. I mean, it's more versatile, but you'll have to do some of your own kind of post-processing as well. So it's not completely hands-free, but it's light years better than what it used to be.
Great. So maybe we'll just take a moment to see if there's any more questions. And maybe in the meantime, I'll just say sort of what's happening next. So at the hour, we're going to have the next tutorial on single-cell transcriptomics that will be led by Sunny Chen. And at 2:45 EST, we'll have another tutorial. I don't actually know how the timing will work. If the first tutorial goes, well, anyway, I'll just sort of speak what the schedule says. So at 2:45, we'll have another tutorial on using single-cell transcriptomics to estimate cellular changes in bulk tissue expression data sets. And then I'm going to go, we're probably going to end this session soon. I'm going to go to GatherTown. Let me put the link to GatherTown in the chat here. And then with that, we'll probably end the session if there aren't more questions. Or let's see, if someone has the link for GatherTown handy, could you post it in the chat? And one more comment is feedback, instructor feedback. Dan, do we have a process in place to get feedback from the students?
I think that there's an email that automatically gets sent out at the end of each day that links to the form, kind of as a prompt. Aaron can correct me if I'm wrong, but I've been seeing that. I think that's the mechanism.
Great. So I'm going to post a link to the KCNI GatherTown. So we're meeting in the KCNI GatherTown, right? And I can safely post that in this chat. Okay, so I'm doing that. I encourage people to try to stop by the GatherTown. It's a great way to meet the instructors and hear more about, have a more personalized interaction with us. I'll be there for a little bit before I have lunch for the next session. Okay, yeah, thanks everyone. It was great talking with you all. Thanks, Dan, for the great explanation of genetics. Thanks, students, for questions. Thanks to our great TAs for answering questions and leading the tutorials this afternoon.
And I will post some of the papers that I promised to post in the Slack space. So if you're looking for those papers specifically on like benchmarking, polygenic scores, some of the approaches to dealing with like multiple p-value thresholds, the giant plus multi-ancestry GWAS studies, I will post those in the Slack.
And Aaron just posted a link for the post-workshop survey. It's very helpful to for us as instructors to get feedback on what worked and what didn't work. We'll probably hold the summer school next year, and it's always great to know what worked and didn't work, and it's good to get feedback on our teaching as well. So Thanks everyone. Thanks for your attention.
Thanks everybody.
See you in GatherTown, and see you this evening or afternoon for the tutorials. Okay, it's evening for some of you. Okay, I will end this session. How do I do that? Yes, end broadcast. Okay, thanks everyone. Bye.